Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Monohybrid cross made easy. Definition, steps, and examples. Mendelian genetics. Table of contents. Introduction to monohybrid cross. Steps of monohybrid cross. Examples of monohybrid cross. Monohybrid cross versus dihybrid cross. 7 differences. Introduction to monohybrid cross. A monohybrid cross is a genetic cross between two individuals with homozygous genotypes for a single trait, typically resulting in offspring with an opposite phenotype. These crosses are commonly used to determine the genotypes of the offspring from homozygous parents. The hybrid produced from a monohybrid cross helps identify the dominant genotype within the alleles. Although monohybrid crosses are often linked to homozygous genotypes, they are also useful in analyzing the genetic composition of individuals with heterozygous genotypes. The success of a monohybrid cross is assessed by evaluating the monohybrid ratio observed in the second-generation offspring. Monohybrid crosses are carried out to determine the dominant allele associated with a particular genetic trait. The cross involves 2 parents, one homozygous for one allele, and the other homozygous for the contrasting allele. This cross results in heterozygous hybrid offspring that express the dominant trait. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Steps of monohybrid cross. Monohybrid crosses are conducted to estimate phenotypic and genotypic ratios and to identify the dominant allele. A specific character or trait is chosen, and alleles are represented using alphabet characters, dominant alleles with uppercase letters, and recessive alleles with lowercase letters. A Punnett Square is created by noting the phenotype and genotype of the parent organisms involved in the cross. The genotype of the gametes is identified, considering that gametes are haploid due to meiotic division. All possible combinations of the genotypes are filled into the Punnett Square, since fertilization occurs randomly. Phenotypic and genotypic ratios of the resulting offspring are calculated and recorded. The offspring produced from this cross are referred to as the F1 generation. Examples of monohybrid cross. First, Mendel's peas. George Mendel used monohybrid crosses to study dominant and recessive traits in pea plants. One example involved plant height, where some pea plants were tall and others were short. The tall pea plant had a homozygous genotype represented as TT, while the short or dwarf plant had a homozygous genotype TT. Crossing these 2 plants resulted in heterozygous offspring with the genotype TT. Phenotypically, the resulting plants were tall, indicating that the tall trait is dominant and the short trait is recessive. Second, Huntington's disease. Huntington's disease is a genetic disorder caused by the Huntington gene. Research was conducted to identify the genotype responsible for the disease. When a homozygous dominant individual was crossed with a homozygous recessive individual, all offspring inherited the dominant allele. It was concluded that the dominant allele is responsible for the disease, meaning all children from such a cross would express the disorder. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Monohybrid cross versus dihybrid cross. 7 differences. Definition: Monohybrid cross. A genetic cross between two individuals with homozygous genotypes for a single character, often producing opposite phenotypes. Dihybrid cross. A genetic cross between individuals with homozygous or heterozygous genotypes for 2 characters. Occurs between monohybrid cross, homozygous individuals with different alleles for 1 trait. Dihybrid cross, homozygous or heterozygous individuals with different alleles for 2 traits. Phenotypic ratio F to generation. Monohybrid, dihybrid. Genotypic ratio F to generation. Monohybrid, dihybrid. Test cross ratio. Monohybrid. Dihybrid. Significance. Monohybrid identifies the dominant allele of a character. Dihybrid analyzes the assortment of traits in offspring. Examples: monohybrid, tall versus dwarf pea plants. Dihybrid, yellow round versus green wrinkled pea seeds. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!